Okay. Okay, the kingdoms. First, you look at kingdoms. Okay, so there are 5 kingdoms: animal, plant, fungi. Okay, so you think of the big picture, then you don't feel so overwhelmed. Okay, so think of the 5 kingdoms: animal, plant, fungi, proto protoctis, and prokaryote. Right? 5 kingdoms, right? Okay. So this one you must remember the way they learn, the way they ask you in IG is this one is multicellular, this one multicellular, this one multi or unicellular, this one is unicellular. Okay? And then the most important is this one got nucleus, right? This one got nucleus, this one got nucleus, this one got nucleus, this one got nucleus. Only this one no nucleus. It's the only one with no nucleus. And then cell wall. This one no cell wall. This one cell wall. This one cell wall. This one sometimes got, sometimes don't have. This one got. Okay? That's the key things you must remember they will ask you. So this one, of course, is like bacteria lah. This one is amoeba. This one is yeast or mushroom. Okay, this one is a plant. Plant is an animal lah. Okay, like a human, dog, cat, whatever, snake. All right, so why their classes so exciting? Our class not so exciting. Oh, Jaehyuk also not here. Did Jaehyuk also go and dance? No, no, no. He's coming. He's just not coming for Saturday on December. Yeah, in December, Saturday he's not coming. He's coming for um Wednesdays. Yeah. Okay.
Oh, Miss Tina, I cannot come to the class on.
Okay.
So what do you have on that Tuesday?
Hmm. Okay. Now, what you must be able to do is most of the question is they will give you a cell and ask you to differentiate. Okay, now the cell wall of plants, yeah, this one got cell wall. This one is made out of cellulose. These are common questions that they will ask you. It's cellulose cell wall. Okay, this one, the cell wall is made of chitin. This one, the cell wall, you don't need to know. This one, the cell wall is neither chitin nor cellulose. It's something different. It's called peptidoglycan, but you don't need to know. You just need to know it's not cellulose. It's not chitin. Okay, can? So bacteria, you must know the structure of it. They have free DNA, and then they have plasmid, and then they got flagellum and got cell wall, cell membrane. Okay, so this is very important. For amoeba, it's a single cell organism. What's the difference is it has a nucleus. Okay, it might be plant-like, it might be animal-like, it doesn't matter. Okay, this one, it's single cell organism as well, but it can have a few organisms combined together. In terms of yeast, they can all bud together and grow together like that. And they can also become as big as a mushroom. Okay? Can? So these are the 5 kingdoms that they will ask you to differentiate. Now, within the 5 kingdoms, plant, they will ask you the difference between flowering and non-flowering. So this one, okay, plants, you will have non-flowering, which are ferns, and ferns reproduce by spores. And then flowering, you have mono and dicotyledon. Remember? Monocotyledon has parallel leaves, long narrow leaves, parallel veins. Dicotyledon broad leaves, branch veins. Okay. Monocotyledon, the flower parts. Monocotyledon, the flower parts are in trees and trees and six. Okay, this one is in fours and fives. All right, can? Do you remember? Okay, now, then after that, for your animals, then you have vertebrate and non-vertebrate. In vertebrate, we have another group. Vertebrate, you have to learn the mammal, bird, fish, reptile, amphibian. Do you remember? Okay. Mammal, fur, mammary glands, external ears. Okay, what are external ears? The ears on the outside. Okay, birds, feathers, wings, lay eggs. Okay, fish, scales, and then of course it has gills. Reptiles and amphibians, this one is scaly skin, this one is smooth skin, and then spend parts of its life in water. Can remember? Okay, now the invertebrate is the one that I think all of you keep forgetting. Invertebrate, there are many groups. The biggest group is called arthropod. All arthropods have jointed limbs. Okay? And then the arthropod, you divide into how many? Four. Arachnid, then crustacean, insect, crustacean, and myriapod. Okay? If I ask you to compare arachnid insect, you cannot say jointed limbs or exoskeleton. All of them have that. All of them have jointed limbs, all of them have exoskeleton. Okay? Arachnid has 8 legs, insects has 6 legs, crustacean has 10 legs, myriapod has 2 in every single segment. This one has 2 segments, head, okay, it's mixed together. Okay, insect has 3 body segments, head, thorax, abdomen. Okay, can? Crustacean, antenna, and more than 10, including the claws. Okay, myriapod, many segments, each segments 2. Can remember? That's for this chapter. Okay, all you needed to count is 1, 2, 3, 4, 5, 6. If you don't know how to count this, we all sit down and cry. Okay? You just have to write 6. Male. Okay? Describe how the 6 chromosome of the male is different from the female. So the female is the set chromosome are the same size. The male is different size. And the male has how many? 3. The female only has okay. Female, 2 chromosomes. Male, 3. Female, both chromosomes same size. Male, different size. Okay, can? So it looks like a very weird question, very difficult, but it's all the same only. Use the word chromosome and gamete in your answer. How variation? Okay, so what happens in the gametes? What process produces gametes? Meiosis produces haploid gametes. Okay, and then the gametes are genetically identical or different from each other. Different from each other. So when gametes fuse together, together randomly. Okay, can you choose which sperm go inside which egg in the wild? No. Okay, there is a new uh, set of chromosomes. Use the words chromosomes. There is a new combination of chromosomes in the offspring. Okay, can you understand? Very beautiful. Does it make sense? Do you understand? Okay. Okay, another cause of variation is new alleles form. Describe how new alleles form. What is the name process? Mutation. Mutation is how new alleles form. Okay. What is mutation? Change in the base sequence of the DNA. Do you remember what is this? Do you know what is base sequence of DNA? The CGTA. Okay, when the CGTA randomly changes, okay, then you can cause a mutation. What can cause this to happen? What causes mutation to happen? So this is mutation. Okay, this is the definition of mutation. How does mutation happen? Ionizing. Mutation is caused by two reasons that we need to remember. Ionizing radiation. Radiation is like, you know how Spider-Man gets a radioactive Spider-Man? Or chemicals. Chemicals that cause mutation is called carcinogenic mutagens. Okay? Ready? Okay. All right. Okay, this is the upper surface of a broad bean leaf. The upper epidermis is there stomata? No. Okay, upper epidermis no stomata unless it's an aquatic plant. Okay. Identify two label structure. Okay, this one's so easy. You label the ones that you actually know lah. Don't go and label the ones that you don't know. Okay, maybe this is nucleus. This looks like the nucleus, right? Okay, but the easiest would be the cell wall and then cytoplasm. And then, of course, if you want here, nucleus lah, can. Okay. State the name of the tissue shown. Upper epidermis, or you can just write epidermis. The tissue is transparent. Why is it important? Okay, to allow light to pass through, to reach the chloroplast in mesophyll cells below. Okay, 3 marks, right? Chloroplast is 1 mark, mesophyll cells below mark, 1 mark, and light to pass through is 1 mark. You can say to trap for photosynthesis, or you can say to trap light energy. Got it? Okay. Stomata are found in the lower surface. The function of stomata is to allow what to go out. This is stomata, right? It allows carbon dioxide and oxygen to go in and out. This one is during the night for respiration, but in the day, oxygen goes out, carbon dioxide goes in for photosynthesis. What else goes in and out? What else goes in and out? Water? Water vapor. Not just water, water vapor. You cannot just say water because water is a liquid. You must mention water vapor. Can you understand? Yes, sir. Okay. You can call this for diffusion of gas or for gas exchange. Okay, that's only 1 mark. All right. What gas do you need? Okay, for photosynthesis, or you can say for respiration. Okay. What else is the function of the stomata? To allow, okay, for photosynthesis, for during photosynthesis, carbon dioxide enters and oxygen leaves. Okay. To allow for transpiration also. Transpiration is the loss of water vapor. Okay. More than 40 years ago, botanists discovered that the stomata will open when the gut sac was turgid. Okay. You know why or not? When it's flaccid, the gut cell becomes like this because it has double layer. When it's turgid, it becomes like this. So when it's turgid, here is the stomata will open. Okay. More than some measurements. Okay. Ions move into gut cells by active transport. How the ions move. So you have to describe active transport. So ions move against concentration gradient. And then using energy from respiration. Okay. Describe and explain the change in ion concentration that causes the gut cell volume to change. Hi, so much to write. Okay, so when it becomes, when the concentration increases, now this is high water potential or low water potential?
No.
Low water potential because more salt, less water. This one is saltier. So when it's saltier, it has more salt, less water. Get it? Okay, so if I had an iron concentration of 0.0, it is low water potential lah. Make sense? Okay, so because it's low water potential, the water from this much become this much. Because water went in or water went out, water went in. Get it? Okay. So the higher the iron concentration, the larger the volume of gut cell. That's one mark. Why? Because higher ion concentration lowers the water potential. Okay, it lowers the water potential in the cell and then causes water to enter cells by osmosis through semi-permeable membrane. Okay, okay, more water in cell, higher Turgor pressure. Turgor pressure, it means the pressure of the cell pushing onto the cell wall. Okay, do you understand? Yes. Okay.
Miss Dana, what's the third line?
High causes water to enter cells, not end cells, sorry. Botanists left the broad bean plants unattended for 3 days. During this time, broad bean plants wilted. Two environmented, okay, high temperature, lack of water. Okay, take a 2 minute. Let's discuss before you're faint. Okay, producer, you can say algae or anybody else that's on the first trophic level. Okay, secondary consumer, you can say the stone crab or the mysid shrimp or the blenny eagle or the marsh rat, rice rat. Okay, feed on two, the bald eagle or the blenny or the spotted sandpiper. Okay, do you know what it means by feed on two trophic levels? That's right. Okay. Okay, this is the nitrogen cycle. So nitrogen in the air, nitrogen fixing, nitrogen in the air, nitrogen fixing to nitrates. This one absorb to become plants. Okay, synthesize protein in plants, protein in dead plants, death. Duh. This one is decomposition. All right. Say the name of process A. Ammonium to nitric ions. Anything to make a nitric ion is called nitrification. And who does nitrification? Bacteria. You don't need to know the name of it. Okay. Describe how nitrate ions enter the roots. Okay, this one you don't know, we sit down and cry. Because we literally just did it. How does nitrate ions enter the roots? By active transport. By the root hair cells. Okay? Across a cell membrane against concentration gradient and using energy from cell. Did you all get all that? Okay, how many marks is this? 3 marks. More than enough. Okay, you can say by protein carriers also and using protein carriers. Okay, any three. All right. Okay, name the structure of plant which process D occurs. Process D is making protein. Which part of your body makes protein? The ribosome. Okay, process that occurs in B. What is B? Nitrogen fixing.
What's nitrogen fixing?
Nitrogen fixing is the nitrogen in the air. Nitrogen N2 molecule is changed to a nitrate molecule.
Haber process.
Haber process is fake nitrogen fixing.
Ah.
It's artificially done because this real nitrogen fixing is too slow to make fertilizer. So nitrogen fixing is basically you take the nitrogen and you fix it into an ion. It can be ammonium ion also. Actually, it's nitrogen to ammonium, ammonium to nitrate. Nitrogen,
Nitrogen to ammonium to nitrate.
nitrogen to NH3 or NH4, then like that. But sometimes from here to here also can. Depends which one you're
It's
talking
not
about.
also for Haber process.
No, Haber process is this to this. Haber process got no bacteria, so we cannot guess. We just add nitrogen and hydrogen in proportion and then it will just react. Get it? Okay. Pyramid of numbers in a wetland shows there were very large numbers of organisms at the base and the pyramid at the top are very few. Okay, we already explained this, okay, about how energy is lost between trophic levels and then only 10% of energy is transferred from one trophic level to the next. How does the energy lost? By respiration, lost by excretion, correct or not? Okay, so only 10% of energy transferred between trophic levels. Most energy lost between trophic levels by respiration and excretion. Okay, what else? Not all parts of an organism can be eaten. For example, I told you before, you cannot eat the chicken feather. Okay, can be eaten or digested. Okay, fair. Okay. So the alternate version of a gene that's an allele. So IA is allele to IO. You know, like blood type, we have IA, IO, IB. IA is an allele to IO. IO is an allele to IB. They're all versions of the same gene.
Yeah.
They're all control blood type. That's what it means.
Oh.
Okay. All right. Mutation in DNA. How mutation results in an abnormal enzyme. Okay, so mutation is a change in the change in the sequence, same as now, of the DNA. Okay, so uh it's a 3 mark question. Okay, so because there's a change in the sequence of DNA, what will happen?
Different.
Different what? Different the active site, this site that is made is same shape or different shape?
Different.
Different. Okay, so the shape of the active site is different, and so the substrate can no longer fit into the active site. Get it? So that's why it does not work anymore. Okay. Can you see? Okay, two tall sunflowers will cross. So if it's tall, it means if the offspring is dwarf, that means it's this lah. So this and this, when you cross together, is 25%. How two tall parents? So both parents are heterozygous. Do you remember what heterozygous means? Okay, so the dwarfism gene, the dwarf gene is recessive. Okay, so both parents are big T, small t. Okay, the figure shows how several strawberry plants can be formed from one parent plant. This is what, asexual or sexual reproduction?
Asexual.
Asexual. Okay, explain why is it asexual. Is there any, is there one parent or two parents? Okay, there is only one parent. Okay, and then uh, they are all genetically identical. All offspring genetically identical to parents, to parent, yeah, and each other. Okay, and then there is only mitosis takes place. There is no meiosis. Okay, disadvantage. Oh, this one we talked about many times already. So there is no genetic variation. So cannot adapt to changes in the environment. That means if suddenly there is a winter that's really long, everybody dies. Yeah, not everybody, all the strawberry plants lah. Changes in the environment. Okay, what else?
More vulnerable to disease.
Yes, okay. More susceptible to diseases. Okay? Yes, done. Correct. Can you all please finish this as homework?
Alright.
Yeah, finish everything as